So we ended talking about proteins, but we had a few more slides. So I want to reiterate with proteins, shape is everything, right? Structure equals the function. The shape that the protein takes will determine how it functions in a cell. All right, so proteins are very diverse, right, in that these 20 amino acids can make up many different combinations of those 20 amino acids to create different molecules, but also they can make different shapes. Right? So here we have an image of these globular proteins, and they're shaped in such a way that they fit together. Right? So therefore their shape is very important to how they function because they have to fit together to perform their function. We'll see later on in the lecture some proteins that are not globular like this, but rather long and thin. Right? Again, long and thin, and they have a different function because of that shape. Right, so structure equals function. You're going to hear me say that not only for proteins, but for so many other things that we'll talk about. If that structure gets disrupted in any way, right? So if this structure starts to unravel, we call this denaturing, right? So if it's being denatured, it's unraveling. It's unraveling, it's losing its 3D shape, and a shape really dictates function. That means that it can no longer function in the way it's supposed to. A lot of things can lead to a shape being denatured for proteins, such as pH. Different pH levels can denature our proteins. Salts can interact with the amino acids and start denaturing our proteins, as well as temperatures, particularly high temperatures. We get really intense heat. That in turn will help to denature our protein. Right, so again, just kind of reiterating that shape equals our function. All right, those are my last two slides on proteins, but we still have one biomolecule that we have not learned yet, and these are our nucleic acids. Now I'm going to give you a broad overview of nucleic acids today, but in a few weeks we'll talk about like transcription and translation. We will review these and go into much more depth on their structure. Nucleic acids are these very large molecules, DNA and RNA. Right? They are composed of monomers called nucleotides. So these nucleotides put one upon each other, creates the strands of the DNA and the single strand of the RNA. Right? DNA, or deoxyribonucleic acid, is double-stranded. So we have two polymers bound together by hydrogen bonds. It stays in the nucleus. It's never going to leave the nucleus. It stores our genetic information, our genetic code, right? But it's never going to leave the nucleus. RNA, on the other hand, is synthesized from DNA. Again, we'll talk about that in a few weeks. But RNA, our ribonucleic acid, can leave the nucleus. All right, so it's created from that DNA, and then basically it acts as a messenger to bring that information outside of the nucleus and then eventually into building a protein. Right, so they carry those protein building instructions out of the nucleus. Both made up of nucleotides, though there's slight differences between the two. Now the nucleotides themselves have a very specific structure. And again, I'm never going to ask you to draw out every single bond and every single atom, but I would expect you to know that it's made of a phosphate group, which we see in yellow here. It has a 5-carbon sugar, which we see in blue, and it has a base. Right, this base are either a single or a double ring of carbon attached to the 5-carbon sugar. Phosphates always going to stay the same. Right? The sugar is going to vary whether we're talking about DNA or RNA. And then the nitrogenous bases are also going to vary. Right? We typically have 4, but that can change if we're talking about RNA versus DNA. So phosphate group, sugar, and a nitrogenous base. If we look at some of those differences, right, we have our phosphate group, we have this 5-carbon sugar. Like I said, this can vary depending if we're talking about RNA or DNA. In RNA, it's going to be this ribose sugar. In DNA, it's going to be a deoxyribose sugar. They look almost identical, except for in DNA, this deoxyribose is missing an oxygen that the ribose has. Right? So very, very similar, very slight difference though. All right, so we have our phosphate group, we have our sugar, we also have our nitrogenous bases, which we can broadly classify into 2 different categories. We can say they're either a pyrimidine or a purine, based on the number of carbon rings that they have. If they only have 1 carbon ring, it's a pyrimidine. If they have 2 carbon rings, it's a purine. Purines are guanine and adenine. Pyrimidines in DNA are cytosine and thymine. Now in RNA doesn't actually use thymine. Instead, it's going to substitute it for a different base, which is the uracil. Right? So in RNA, we have 2 differences. We have uracil instead of thymine, and a ribose instead of a deoxyribose. So a good question I could ask on an exam would be something like, this molecule has a ribose in it. Is it RNA or DNA? Right, that'd be a fairly simple question. Now, like carbohydrates, like proteins, we're going to stack those monomers upon each other to create a polymer. The polymer, in this case, we would call nucleic acids. Right? They're the DNA, they're the RNA. But we're going to stack those nucleotides upon one another in what's called a sugar phosphate backbone. Right? So in red here, or in this image as a whole, we have a DNA strand, just one strand. DNA typically has 2 strands. In red, we have the sugar phosphate backbone. Right? Now, it's the sugars and the phosphate that link the nucleotides together. Nitrogenous bases, right, just kind of hang out towards the center. So if we look at this backbone, we have a phosphate and a sugar. And a phosphate, sugar, phosphate, sugar, phosphate, sugar. If we were to zoom in on that sugar, we can label or number the number of carbons in that 5-carbon sugar. We can label the carbons. And so we might start on the right-hand side here. So we would say that's carbon number 1, or the 1' carbon, 2' carbon, 3' carbon. Other side, we have the 4' carbon, and coming off of the 4' carbon, we have the 5' carbon. Right. Now, in the sugar phosphate bone, we're always going to link the 3' carbon to the 5' carbon. So 3 and the 5 will be linked together with a phosphate group in the middle. All right, so if we go back to this image here, we have our carbons labeled here, 1, 2, 3, 4, 5. You see we have a phosphate group coming off the 5, right, and a phosphate group coming off the 3. So essentially we have this 3' linked to a phosphate, linked to the 5', same molecule, the 3' linked to the phosphate, then linked to the 5' of the other molecule, and so on. Right? Now, excuse me, just like we've seen in the past, it's going to be a dehydration reaction. Right? So if we take a closer look at this, the 3' has an OH. All right, the phosphate group also has an OH. We take 1 oxygen and 2 hydrogens away, we get a molecule of water, and what's left is that covalent bond, which, just like our glycosidic bonds and our peptide bonds, has a very specific name. We would call it a phosphodiester bond or a phosphodiester linkage. All right, so dehydration reaction yet again, linking our 3' to the phosphate. That phosphate is linked to the 5' of the next nucleotide. All right, so we have our sugar phosphate backbone, but what is the role of our bases? Well, our bases are going to face inwards from our strands, and the bases will actually become bonded together via hydrogen bonds. And it's these hydrogen bonds that helps give DNA its classic double helix shape. So if we look here, adenine is always going to bind to thymine. And if we see in the middle here, those dotted lines, those are indicating those hydrogen bonds. Same with cytosine and guanine. Right? In this case, we have 3 hydrogen bonds. So not only do they play a role in the structure of the DNA and creating that double helix with those hydrogen bonds, they're also going to encode all of our genetic information, which we'll get into later in this semester. All right, let's review the last 3 lectures we've been talking about these biomolecules. So for each molecule, I want you to determine, does it have a monomer? And if it has a monomer, what is the name of the monomer? And what is the name of the polymer if we put many monomers together? All right, I'll give you a few minutes. Yeah. right, so our review. I want thumbs up if it has a monomer, thumbs down if it doesn't. Carbohydrates, do they have a monomer? Yes, I like it. What is the monomer's name? Monosaccharides. If we put many monosaccharides together, we get polysaccharide. Yes. What about lipids? Thumbs up or thumbs down? Thumbs down. Yeah, it does not have a monomer. Right? Even though they're made up of 3 fatty acids, right, and a glycerol, if we're talking about fat, right, they just exist on their own. They're not going to be repeated over and over and over to form huge molecules. So they do not have monomers. Rather, we're talking about a phospholipid, a fat, or a steroid. What about proteins? Thumbs up, thumbs down. They do, yes. And what would we call monomers? Amino acids. If they all join together, we would form a polypeptide. And the one we just talked about. What is, oh yes, thumbs up, thumbs down. Yes, it has monomers. What is the name of the monomer? Nucleotides. The name of the polymer? Nucleic acid, because it's in RNA. Yeah, very nice. All right, any questions on biomolecules? We are going to completely change subject now. Maybe we're tired of talking about biomolecules. So we're going to start talking about the cell. So we're going up in our level of organization. We went from atoms to molecules. Now we're talking about cells. All right, so if we're talking about cells, we can broadly classify cells into 2 different types. So 2 different categories of life. We can be a prokaryote, we can be a eukaryote. Right? Prokaryotes, they're very small. Typically, they're only one cell for the organism, there's only one cell itself. And they don't have a nucleus. Versus eukaryotes, which would include organisms like ourselves and the plants we see outside. Right? These cells are much more complex. They're a lot larger. They have a nucleus. The nucleus contains our DNA. But they also have a lot of different other compartments within the cell that we would call organelles. And we'll go over the organelles in a bit. Right, the prokaryotes, fairly simple, no nucleus. Eukaryotes, more complex, has a nucleus. We're not going to spend too much time on prokaryotes, but I did want to go just a little further to say that prokaryotes can further be divided into 2 categories. I always think I have this slide in here. So this slide is a great chart to kind of tell you the difference between the 2. We've already mentioned it, right? Eukaryotes, typically larger. Eukaryotes have a nucleus. Eukaryotes have many organelles. Cytoskeleton, we're going to ignore this line for now because we will talk about the cytoskeleton in a bit. Right? So this is a good review slide as you're studying. All right, but like I mentioned, prokaryotes can be further divided into 2 different types. We can say they're bacteria cells or archaea cells. Both are similar in size in that they're both very small. Right? They are tiny cells, from 1 micron to 10 microns in diameter. Bacteria, however, are very abundant. We find bacteria everywhere, right? Science has not stopped discovering different species of bacteria. There's so many out there that we just haven't named them all. Now, the vast majority of these are not harmful to us in any way. In fact, we might have symbiosis with a lot of these bacteria. We have our gut microbiome. Our microbiome is made of bacterial cells. Right? If that gut microbiome gets messed up, we might feel really sick. Right? They perform a lot of important regulatory functions. There are some species that do cause diseases, though, right? Which you might need to go to a doctor and they might prescribe you antibiotics, which treat that specific bacteria. But the vast majority of them, very benign. In fact, we could not live without bacteria, so we need bacteria. Archaea, on the other hand, much less common, and we really only find them in really hostile environments. Think about like deep in the ice in Antarctica, or in the geysers of Yellowstone, where it's like extremely hot. Right? They're very hardy cells that can survive these really hostile environments. We're not going to talk too much about archaea, but I am going to elaborate on bacteria a little bit more. Specifically on the shape of the cell itself. So if we look at the structure of a bacteria cell, it's fairly simple. Right? We have our cell, right? Inside of our cell, right, we have the fluid of the cell that's surrounded by our plasma membrane. We also have plasma membranes in eukaryotes. We don't have that nucleus, but instead we have a region where the DNA is found. We would call that the nucleoid. Bacteria cells can also have a cell wall, which is kind of unique. The only other types of cells that typically have cell walls in eukaryotes are plant cells and some fungal cells. Right? Animal cells do not have a cell wall. They're going to be coated in this glycocalyx. It's like this outer gelatinous covering. Think of if you dip the cell in mucus. All right, that's kind of the consistency that we would be thinking of here. Can protect it from many environments, allow it to live in different environments. They also tend to have these extensions coming off of the cell, either pili, which are short extensions, or flagella, which are a lot longer. Now, these are not exclusive to bacteria cells, but bacteria cells tend to have these more often than not. They also have one type of organelle, and we'll describe this organelle in a little bit more detail in a few slides, but they have ribosomes, right, which synthesizes polypeptides, synthesizes proteins. But that's really the only organelle that they would commonly have. They don't have any of the others. All right, that's vastly different from our eukaryotic cells. All right, so our eukaryotic cells much more complex, and is the reason that we are going to spend the rest of our time in lecture today talking about only eukaryotic cells. So eukaryotic cells, we have a nucleus that contains the DNA. And if we remember, the DNA does not leave the nucleus. It always stays in there. It'll make RNA, and that RNA can leave the nucleus, right? But the DNA stays in the center. We also have many, many organelles. So organelles, we're going to broadly define for now as being these compartmentalized membrane-bound compartments. They each have a specific function. Right? You can have organelles like the nucleus, for example, and the ribosome, but also the endoplasmic reticulum and the lysosome and the Golgi apparatus, which we'll talk about. Now, eukaryotic cells are very diverse, right? We don't just have one type of eukaryotic cell. We have a whole variety of them, right? So a really good example of this is within our own body. If we took a neuron from our brain, a neuron cell, and we took a epithelial cell from our skin, they're going to look different, right? So a neuron typically looks like, I have to think of it as like a hand spreading its fingers, right? Epithelial cells are more circular or cylinder shaped, right? They're going to have DNA in their nucleus, right? But they might contain different organelles, different amounts of those organelles, right? They all have maybe a different shape like we see with the neuron there. We can also see the difference between species, right? And really the big one here is between plants and animals. Those cells tend to look a little different. They still have a nucleus, they still have organelles, but there's a few organelles that a plant has that the animal cells does not, and vice versa. So lots and lots of organelles, lots of variety here. If we take a look at how plant cells and animal cells differ, this is a very broad overview. I'm actually going to have you do an assignment that looks into this with a little bit more detail here. But we see the plant cells have chloroplasts and cell walls that the animal cells do not. Likewise, the animal cells have centrioles, which are important in cell division. All right, so when we talk about mitosis and meiosis, we'll start talking about centrioles. They also tend to have different vacuoles, different sized vacuoles. So the assignment that you'll do will go over this in more detail. All right, let's review what we know so far. Right, pretty much the only time we'll talk about prokaryotic cells. What is the main difference between prokaryotic and eukaryotic cells? What are those similarities? Yes, if you ever need to step out for that, just go ahead. Okay.
Well, the prokaryotic.
Okay, it was on campus.
Go back and look at the slides.
Who wrote more than you? They would know.
Feeling confident in what we know so far? This is review.
There we go.
This is one of those lectures where you've probably heard of most of the concepts, but I still have to review.
We definitely didn't really talk a ton about prokaryotic and eukaryotic cells in my class.
Which is pretty common, unless you take a course dedicated to prokaryotes. Likely won't talk about them. Or if you take organismal biology, there's a small section dedicated to them.
Like I knew that there were something, but I didn't know what they were, what they did.
Yeah.
Yeah. The prokaryotes, like bacteria.
Good. Review.
It's easy.
All right, what are some of those main differences between prokaryotic and eukaryotic cells? One of them has a nucleus, one of them doesn't. Yes, that's a really main difference between the two. What else could we say is different? Yeah, one's very simple. Prokaryotic cells are simple cells. Eukaryotic cells are very complex. They contain more. Anything else for differences?
Eukaryotic cells are a lot more organelles than prokaryotic cells.
Yes, a lot more organelles. So even though prokaryotic cells might have organelles, eukaryotic have a ton more.
That the prokaryotic cells typically have cell walls.
Yeah, they have cell walls. And we kind of see those in some of our eukaryotic cells, but not all of them. All right, what are some similarities? How are they similar? Yeah?
Both have DNA.
They both have DNA. The location of that DNA is different, but they have DNA. What else?
They both have ribosomes.
They both have ribosomes, which aid in making proteins. Any other similarities we want to mention?
There's a plasma membrane.
Yeah, they have a plasma membrane. Yeah, those are all really good similarities. We could say they also have cellular fluid on the inside. We could say that flagella and the cilia, we have some eukaryotic cells that have those as well, right? I think we touched upon all the main ones here. So let's move on to continue with our eukaryotic cell. Right? So there's two terms I want to make sure that we have down before we go on to the rest of the lecture here. And those two terms are cytosol and cytoplasm. Right? Cytosol really refers to the cellular fluid. So it's everything inside of the plasma membrane of the cell, but outside of the organelles. Right? Versus cytoplasm means everything inside the cell. Organelles and the cellular fluid alike. Right? So the cytosol is part of the cytoplasm, but the cytoplasm is not part of the cytosol. We're going to go on to look at the cytoplasm, so everything inside of the cell, in a little bit more detail. We'll talk about the cytoskeleton, we'll talk about the endomembrane system, and we'll talk about the semi-autonomous organelles. I know we have a lot of football players, so good luck on your scrimmage
Thank you.
today.
Have a good day.
Good luck.
Yeah, you as well.
Hey, Walker.
Thank you.
All right, so cytosol and cytoplasm. We're going to start by looking at the cytoskeleton. I feel like the cytoskeleton is the most often overlooked thing inside of the cell. We often, when we see pictures of a cell, the cytoskeleton is not drawn in, just for sake of simplicity. But the cytoskeleton is this network of filaments that run throughout the cell. And we can have different types of filaments, right? Different shapes of the proteins, again, that have different functions. So now we're looking at proteins that are really long and thin. Right? The microtubules are the thickest of the three. They tend to be hollow on the inside, and they're more or less unstable. Unstable? I probably should say unstable. We also have our intermediate filaments that are rope-like. So if you think of rope, it's made up of many different threads, all kind of woven together to create a rope. Same concept for our intermediate filaments. And then we have our actin filaments, typically the smallest of our filaments. Right? They're also known as microfilaments. All these types of filaments work together to help the cell hold its shape, to help organize the organelles and molecules within the cell, and to help move those molecules and sometimes organelles within the cell. So the cytoskeleton really helps it hold its shape. But when we think about organization and movement, now we have to think about motor proteins. So these are yet another type of protein that helps move cargo throughout the cell. Now that cargo can vary. It can be molecules. It might be rearranging some of the organelles, right? They're all going to need energy. So they're going to use ATP as its energy source. Right? And they typically have three different domains: a head, a hinge, and a tail. Right? And responsible for a lot of the movement inside of our cell. So if we put this into an analogy, if we're walking, right, we would say the ground is our filament, the cytoskeletal filament. My leg is the head of the motor protein, and my hip is the hinge. So I'm just kind of walking along my cytoskeleton filament here. Many different types of motor proteins. I don't expect you to know all the different ones. If you take a proteins class, then maybe you might be expected to know all those. Now these motor proteins, I think they're kind of hard to conceptualize, so I'm going to show us this video. I think it's a cute video that kind of helps you understand the cytoskeleton, the motor proteins, and how they all work together. Now this video is going to talk about it in terms of a neuron cell, but know that this happens in all the cells. All right, so I really like the analogy that it makes there where the cytoskeleton are the roads, like the major highways, down to the alleyways here. I'm going to brighten it back up so you don't fall asleep on a Monday morning. All right, and I like the way that it shows the movement, that walking analogy. Right? Now that's one type of movement, and I just wanted to mention a couple other types of movements that can occur. So in that movement, that motor protein is carrying the cargo along the filament. The motor protein is moving. There can be another type of movement where that motor protein doesn't move. It remains in place. And the filament, instead, is the one that moves, causing the motor protein to move. So if you were to think of my hand as the protein and my arm as the filament, right? My hand is not going to move, but if I were to move my arm, it moves that motor protein. The same thing is happening here. We often see this a lot in like muscle movement. The third type of movement is one where both the motor protein and the filament is stationary, but rather there's a force exerted by the motor protein. So if we have two filaments and the motor proteins would be on the inside here, there would be a force exerted by the motor protein which causes the filaments to bend. Right? So it's this bending movement now rather than a walking movement or like that hinge movement for when only the actin filament moves. Right? We often see this bending movement more often when we're talking about like cilia and flagella. Right? So those thin protrusions off of the cell. So flagella tend to be a lot longer than the cilia. They can be present just one or in pairs. The cilia are often shorter and cover the entire surface of the cell, but they all kind of move in this bending motion, which is where that force is being exerted on the filament. All right, question for the cytoskeleton review here. So I found this image on the internet. I don't know about you, but I've been seeing a lot of those giant skeletons popping up everywhere now that we're closer to Halloween. So I want you to look at that image and tell me in which ways is the statement accurate and in which ways is it inaccurate. And I'll give you a few minutes. Are we understanding cytoskeleton? Some people have them. I think it's kind of like mental. It's something you should talk about in this lecture.
Yeah. Yeah, I know. I was thinking, I was talking so much shit, I got my, bomb. And then I stepped in, you go, you just hold vengeance, and I got them. I'm just so I haven't read their role for it. Because I know what they tell Is it really looking like that's so smart.
All right, I feel like everyone's answered this question. Maybe 3 minutes was a little long for this review, but that gives you time to catch up on your notes if you need to. So, this image, in which ways is the statement that the cytoskeleton is just chilling, keeping the entire cell together? In which ways is this accurate?
It holds the body together, similar to the cytoskeleton.
Yeah. It's the skeleton of the body, just like the cytoskeleton is the skeleton of the cell. It's holding it together. Any other ways in which this is accurate? I think there's more ways in which it's inaccurate. So, how is it inaccurate?
The part when it's just chilling, it's like holding the entire cell, it's gonna like move in a lot of ways, so it's gonna be like tired out.
yeah. If it's just chilling, right? It does a lot of things, so maybe it gets tired out. Yeah, I like that analogy. But there's so much more going on, right?
Yeah, it's not just the cytoskeleton, it's a holding the cell together. So there's for some cells, there's a cell wall and other things like that.
Yeah.
Structure.
Yeah, it's not just the cytoskeleton holding the cell together. I like it. So just think of like the cytoskeleton, it's not just the filaments, but it's also the motor proteins. It's also all the movement that's happening within this cell. Right? It's not stationary and chill. All right, any questions about the cytoskeleton? We're going to move on to maybe parts of the cell that you're more familiar with. And so these would be like the organelles and how the organelles work together and what functions they perform. We would call it the endomembrane system. Right? So all those organelles inside of the cell, how they're connected, either physically or functionally. Right? Many of these organelles will see that their functions actually work together with other organelles. So I'm going to go through a list of organelles here. Right? Many different of the main organelles that we're going to see. I like this image. It kind of shows you plants versus animal cells here. Though it is missing the vacuole in an animal cell, which is there. It's just really small. So I'm going to go through this list, and I'm going to try to go relatively quick, but I'll still try to speak slow, just because this is probably stuff you've read about, learned about in biology classes in high school, and have an idea of what happens. So, for example, the nucleus. We've already talked about the nucleus, right? It contains the DNA, right? The DNA is the code by which we would produce proteins. So the nucleus contains the DNA and will direct protein synthesis, right? It makes the RNA. The RNA can leave the nucleus. Think of the RNA as kind of like if you were mailing a letter, right, containing a code. That letter is the RNA moving to its next location before it's decoded. All right, so our nucleus contains our genetic material. Our ribosomes then, kind of continuing on with the nucleus, how the nucleus made the RNA is leaving, the RNA is going to end up at a ribosome. And it's the ribosome's job to decode the RNA and make a protein. All right, so ribosomes are involved in protein synthesis. We might see some cells have a lot of ribosomes, particularly if that cell has to make a lot of different proteins. Other cells, maybe they have fewer ribosomes. Think of maybe like your bone cells, right? They're more structural than making proteins. We can find ribosomes just kind of hanging out on their own, or we can find them embedded in the endoplasmic reticulum. So the endoplasmic reticulum is the one that we'll talk about next. This organelle can either be rough or smooth. Rough if it's embedded with ribosomes, smooth if it lacks ribosomes. So the rough ER, it's like a network of membranes, like these folds upon folds of membranes. The rough ER, in conjunction with ribosomes, is important for synthesizing proteins, right? So together, they will synthesize a protein. Now the smooth ER doesn't have any ribosomes, is not important in protein synthesis, but rather in lipid synthesis. So oils and steroids and phospholipids. It also plays a role in helping to store calcium ions. Right, so rough, we have ribosomes, we have protein synthesis. Smooth, we're lacking ribosomes, and instead we're focusing on lipid synthesis. Right, and together we're just looking at like this whole network of membranes. I want to zoom in on the rough ER a bit on this next slide so you can have a visual of what it looks like when a ribosome works together with the rough endoplasmic reticulum. So we have our ribosome here. Right? It's kind of clinging to the outside of the membrane of the rough ER. The ribosome, right, it's receiving the RNA, right, receiving the instructions for a protein, and then actually making the protein. But as it makes those proteins, as it adds these different amino acids end on end on end, it's secreting that protein essentially into the rough ER. Inside the rough ER, that's where the protein will get its different levels of structure beyond the secondary or beyond the primary structure. It'll get the secondary structure, the tertiary structure, the quaternary structure. It'll get its 3D shape. Once that protein is fully made, then the endoplasmic reticulum can essentially transport that protein to where it's needed, which might be another organelle for further processing, or it might be a part of the cell where it's going to end up. All right, so we're working together. These organelles are working together here. Now, if you were to look at a cell, you likely would see two different types of folds of membranes here. Often they're color-coded in our images. Because we also have the Golgi apparatus. The Golgi apparatus is also a stack of flattened cells, right? Flattened membranes, right? So these membrane-bound compartments stacked upon one each other. And the Golgi apparatus plays three main roles: secretion, processing, and protein sorting. So sometimes those proteins will come from the endoplasmic reticulum and end up in the Golgi apparatus, where it's further determined where it needs to go next. If the protein ends up in the Golgi apparatus, sometimes the protein is further processed. Maybe it's adding some carbohydrates to the amino acids within that protein to give it its specific function. It can also secrete materials. We can see in this image here, we have this cargo and a vesicle. All right, it's being secreted out. So basically part of the plasma membrane is folding up on itself, creating a little sphere containing our cargo. It's going to kind of join with the plasma membrane of the outside of the cell and release its cargo to the outside. Right, so they don't have the most exciting of functions, but they help with a lot of regulation within the cell. All right, a few more organelles to get through here, one of which are lysosomes. So lysosomes aid in digestion or in breaking down of really complex molecules, right? Inside of our lysosomes, we're going to have the process of hydrolysis, right? So adding water to break down these larger polymers. And in fact, they contain a protein to help move this process along even faster that we would call an acid hydrolase, right? It's an enzyme that really helps make this process a little faster. The lysosomes, right, they contain different types of these enzymes depending on the different types of molecules they're breaking down. And they're formed from the Golgi apparatus. Right, this is different from our next one, peroxisomes. Right, so peroxisomes also function in breaking down molecules, but they break down different molecules using a different enzyme. Right, so now instead of hydrolysis and acid hydrolases, they contain a different enzyme called oxidases to help break down the molecule. And we find a lot of these in liver cells, right, because they're really good in detoxifying, getting rid of any toxins that might have accumulated inside of our cells. So the lysosomes, they were formed by budding off of the Golgi apparatus. Peroxisomes are formed via the budding off of an endoplasmic reticulum. So different sites of formation as well. All right, my next slide is vacuoles, but my last class we did not get to the vacuole slide. So I'm actually going to skip it and we'll start there next Wednesday. But I want to mention we have 2 homework assignments that I'm introducing, one of which is due on Wednesday. The other one is this content check. So the content check number 2 is up. It's not due till next Monday. I really want you to print it out and write your answers, or type your answers and upload it digitally. What I don't want you to do is write out the questions on notebook paper and then write out the answers as well. It's more work on your end and it's harder for me to grade. And then the other assignment is for Wednesday. This one is worth points. I'm having you watch 2 videos, answer 2 questions, upload it as a PDF on Moodle. If you type your answers and print it, that works as well. I would encourage you to upload it just because it saves on your printing. And that's all I got for you. So, enjoy the rest of your Monday.
Oh, there's questions.